All of you are familiar with mayonnaise. All of you should know there are millions of droplets of oil in water in every ounce of mayonnaise. We use that same concept, but this time inverted. Instead of oil in water, we have droplets of water in oil. When you have that, you have absolutely separate drops. Those drops are as separate as test tubes or plates. And what we did is take technology that had also won the Nobel Prize, polymerase chain reaction, and instead of doing it in a test tube, we did that in individual droplets. Now, one of the keys to this was stabilizing the droplets, and there were some brilliant scientists at 454 who came up with an additive from the explosive industry which allowed them to stabilize, they all got on watch lists, of course, stabilized these droplets in the tube as it went through cycling. And so you would break up a genome, you'd have one piece of Jim Watson in one, another piece on another, and after 30 cycles, each time doubling a copy of just that fragment, you'd have a piece of Jim Watson uniquely duplicated in each of millions upon millions of wells of beads. So instead of lots of robotics and years of prep and bacteria, the whole thing can be done in a test tube. And it works. Again, the second half of the problem is taking those individual pieces of DNA and decoding those letters, the A, C, G, T, the equivalent of a computer program's ones and zeros, the blueprint for your genome. And you would take those individual beads, and instead of running sequence on large machines in big facilities, to point out this is a big facility at MIT where they can spend lots of money, and you have these beautiful little 454 machines here that... That's ours. Oh, you switched the pictures on me. Okay. Good. So instead of having a large facility, you could have a single plate that would replace it. And now one person, instead of a team, could really do genomes at a time. Now, one of the important things about a technology is really the difference between an invention, and I like claiming I am the inventor of 454 sequencing, and an innovation. An innovation is taking an invention and making it practical for others to use. And that's really what Michael Agholm, Srini, and other people did at 454. And they were able to make this so practical that they could deliver machines to researchers around the globe, and each of those researchers could do their work. And that's the key. So this is just an example of publications that are the result of going from an invention to an innovation to scientists using it to solve problems that they faced. Like any technology, when it's new, you consider it inferior. And when we first published our technology in 2005, we were able to do 20 million letters or bases at a time. That was about 100 times faster than anything before us, but it wasn't as good in terms of its quality. So again, just like the first time, the cynics said, you know, but that won't work for real genomes like humans. But again, Michael and his team at 454 were able to take that initial technology, make it 5 times better in 2 years, and increase its accuracy, if you will, over 20-fold. So now it would be 99.5% accurate and produce 100 million bases. Now, people have a different way to have a perspective on 100 million bases. When I was getting my PhD, I sequenced over 6 years 10,000 bases, and I was quite proud of that, but I had a colleague, Tian Chu, who's now the vice chairman of human genetics at Yale, and he didn't think you should get a PhD until you sequenced 40,000. So I was quite devastated at that moment. But the point was, it took 6 years for one person to produce 10,000 bases of sequence. And now one person in 7 hours could produce 100 million. What would this enable? Well, from the perspective of myself and 454, we collaborated with an amazing scientist, Svante Pabo, and with Svante we initiated the Neanderthal Genome Project. We would do something that was impossible. We would take DNA from a Neanderthal that was 38,000 years old, sequence it, and over the next 2 years, put it together.